Nature Works podcast. Conversations with extraordinary guests who are working to protect, regenerate, and better understand the natural world. With your host, Mike Weeks.
Welcome to Nature Works podcast, in which this week I'm speaking with Mike Allen, PhD. Mike's an associate professor of single-cell genomics. Don't worry, he does explain what that means. He's based in the College of Life and Environmental Sciences at the University of Exeter. And in this episode, Mike schools me on the essentialness of viruses and how all life depends upon them. If you think that killing all viruses is a good idea, then think again. He also talks about how to kill any bacteria, even those that are nuclear proof. Did you know that there are bacteria living on the inside of nuclear reactors? I didn't. And also, Mike's a smart guy, but not as smart as his young daughter, who suggested a revolutionary approach to forming plastics and seaweed together to create a new form of oil. We also discuss Mike's rather hilarious car crash meeting with Bill Gates. And as usual, on top of that, we cramming as much science and as many anecdotes and funny and interesting stories as we possibly can. Now, if you enjoy this episode and others of NatureWorks podcasts, please share it with the folks you know who care about the natural world. NatureWorks podcast aims to provide honest and unbiased insights into how we can help protect, restore, and regenerate the natural world. Thanks for being on. I've heard lots about you, and I've heard not just from the scientific perspective, but the descriptions I've had about you are that you're a little bit maverick, and probably giving that away. And in fact, the lady that recommended that you come on said, you know, he's one of those scientists who doesn't give a fuck about upsetting people. He's just interested in the truth. And I thought that's a good place to start. What does she mean by that?
I have no idea. No, I guess what she meant is I speak my own truth, as it were.
Isn't there only one truth in science?
Oh God, there's a question. Well, I thought you were going to break me in slowly. I guess.
Break you down.
Well, you know, I think we live in a world where, especially with social media, everything's black or white, yes or no, right or wrong, and I mean, life exists in the grey areas in between, and I don't think people really appreciate that about science sometimes, is that they want a yes or no or a right or wrong answer, but actually, you know, it often is in between, and the more information you have, the more informed you are. So that's a crazy first question to break me in on.
Well, there's it's Karl Popov, isn't it? Who's the guy from the 60s or 70s who said that the only way that you can actually prove you can't actually prove something to be correct because you have an unending new proofs. The only way you can actually know if something is correct or not is to disprove it. And yeah, I'm not a scientist, but I spent the last 6 years of my career working with my co-CEO as a scientist, and he's a nerd like you. He doesn't have your flair for shirts, I'll add, but you know, he's a bio. What was it? His is geochemistry, and so I've learned all about the scientific approach very deeply the last 6 years, and come to the same conclusion that everyone's making it up to some just with different sets of variables, you know, and there's a lot of cognitive biases in the scientific world as well. I mean, there's such a thing as Christians for science, isn't there, in America? How does that work?
I mean, crikey, the second question is getting onto religion and science. Brilliant. Okay, all right. So, I mean, I think one thing we're all very good at is compartmentalizing our lives. And, you know, you can have massive sort of contradictions in people's lives. You know, like, well, you look at America now, it's all sort of pro-life, but with all the guns. I mean, it's crazy. Yeah. I mean, they're saving lives so they can be shot at in their schools. I mean, well, it's absolutely nonsensical when you think about it. And I guess from that point of view, when you look at religious approaches and science, maybe that's what scientists who are religious do. They just compartmentalize it. I'm not saying whether it's right or wrong. I mean, I'm an atheist, but you know, I respect people's views, and I think in a world where you can be anything, just try and be nice. I mean, as long as it's not harming anyone, let people believe what they want. So,
yeah, the US right now is like the ultimate kind of display, I think, of a society that has gone so far off the rails that you can pick and choose 1,000 different subsets of its cultures and find something wrong with it at the moment. It's just there's contradictions everywhere, right? Everywhere you look. I lived there for 6 years before I came here to Bali. Bali's so much simpler. People are just getting on with their lives and day to day, it's ceremony, do your work, enjoy, be with your family, and the likes. And so, for me, I don't think I'll be going back into the US any day soon. I didn't mean to hit you straight with religion and politics, and I'm not, which is why I'm not going to go deeper into it, even though they're two of my favourite subjects. But the science piece we do have to go deep into, because you are a proper scientist and you are a single cell genomicist.
Yeah, that's right. That's my official title.
So what the hell is that? What's a single cell genome, and what the hell does a genomicist do?
Oh God, right. That's another cracker. Okay, all right. So to get to that stage and that job title, it took about 20 years, and it's a very meandering path. So I oh God, right. Okay, I probably have to rewind and go all the way back to the beginning.
Well, I was about to say, did when you were 4 years old and were you thinking when I grow up, I want to be a single cell genomicist? Am I even pronouncing it correctly, genomicist?
Yeah, not many people actually say genomicist, so you can say it how you like, because you just sort of,
I'm making it up. Yeah, are you making it up, Mike? Mike, are you even a scientist? Is this all bullshit? Yeah.
No, not. I think we all suffer from imposter syndrome.
Yeah, I saw somebody say that today, actually, that everyone suffers from imposter syndrome, and I went, I bloody well do not. And then I went, that was probably my cover there.
I mean, I think when I moved to Exeter University, one of the first talks I gave was, I basically said, look, I'm an imposter. Consider this talk as therapy. And then I sort of outlined everything I did and how I didn't know what I was doing when I was doing it, but it all made sense in the end. And I think it's looking back, it's really easy to make sense of my career. It's just that when you live it day to day, none of it makes sense. I'm kind of, I've always had 2 rules in my research group. You either do cool or lucrative science, ideally both.
I like that. Those are really good rules of thumb. Because I mean, there's an endless amount of frames or rules of thumb that you could put on the type of science that you're doing, but cool or lucrative. Does that not pigeonhole you though into, I don't know, working only for Tesla or?
No, not at all. Not all cool science doesn't have to be expensive science.
Okay. Yeah, but the lucrative stuff does.
Yeah, the lucrative stuff can support the cool stuff. It provides you with a resource. It's like doing a deal with the devil, isn't it? I'll do your work for you, which may not interest me, but you'll give me the resources to do the work that I can do on the side. So, that's the sort of approach I take.
That's 99% of the employed working world, isn't it?
Yeah, I think so, probably.
All right, so you're not getting off the hook. You're not getting off the hook here, though, because there's people going to be listening, going, no, he didn't answer the question. What the hell is a single cell genomicist? Tell us what a single cell genome is first.
All right, a single cell genome is exactly, break it down. You know what all those words mean, put them together, and it's exactly the same thing. So a single is one on its own. A cell is the smallest unit of life, and a genome is the genome that's contained inside of it. So basically, my job title is officially I'm an associate professor of single cell genomics, and I work on. Oh God, how to say this?
Basically, do you want me to help you? Mike works on genomic, proteomic, transcriptomic, and metabolomic approaches. Yeah. So it's not going to get any better with me, is it? You better help here.
I work to understand how cells work, and when you think about a lot of scientific approaches, when scientists go out and they sample the natural environment, for example, and then they combine everything together, extract compounds from it like DNA or RNA or metabolites, and then they look at them. And then essentially what we're looking at is is an amalgamation of noise of lots of different things. Even if you have a one culture with one type of bacteria or algae or virus in it, you have hundreds, thousands, millions of different types of algae, viruses, and bacteria in it. So by going to the single cell, you actually remove all the noise. So it's like, everything we do when we sort of profile cells and cultures is an average of every cell in there. So if you had a culture of bacteria growing, for example, you'll have some that are dividing, some that have just divided, some that aren't dividing at all, some that are dying, some that are thriving, some that are nutrient limited. Even in a flask that's mixing that looks homogeneous, there's always that sort of natural variation. That's why we do technical replicates and scientific replicates, is to get rid of that noise and average it all out. But with single cell genomics and single cell approaches, you don't have any of that noise. What is. It's the true signal, if you know what I mean.
Yeah, I get that. But is it not the case, though, if you take a cell out from the organism, that it's like the old metaphor of a bunch of blind men trying to tell you what an elephant is? One's got the tail, one's got the trunk. And they both think one thinks it's a skinny snake and the other thinks it's a fat snake.
Yeah, and each of those blind men is right.
But then how do you? But they're right about their part. I'm trying to understand how it works because I've never studied cells like you have.
Okay.
So how does it work from a perspective of you take a cell out of the organism and then how do you?
I, as an environmental microbiologist, most of what I work on is single-celled organisms anyway.
Okay. So that makes it convenient, doesn't it?
It does make it very convenient. But the actual technique itself is applicable to multicellular organisms. So, for example, every cell in your body is not acting the same right now. You have muscle cells that are acting like muscle cells, heart cells like hearts. When you get cells
my liver's working overtime. Yeah,
your liver will be working overtime. People, people suffer from infections, so those cells will be responding differently. People get cancers where the cells go out of control. So what single cell genomics allows you to do is identify individual cells and exactly what they're doing. So, every cell has the right information, similar information to all the other cells, but they're not all acting the same. And that's what single cell genomics allows you to do, is to see what things are acting differently and why. And
you study them under a microscope?
I have microscopes. I use lots of things to study them. A lot of the things I work on viruses, a lot of viruses, and you can't work on those with a light microscope. So we work with those with electron microscopes and possibly the coolest thing I own in my lab, which is an atomic force microscope, which is pretty damn cool.
Let me come on to that, how that works, because I really want to know, and I want to know why you can't use light microscopes. But my question about studying a cell in that form is that if you're, let's discount single cell for a moment, and then we'll come back into that. But if you study a cell outside of the organism, does it behave differently than when it's in the organism? Is it not a bit lost? It's not like separating it from its pack.
So in the context of your question, when you say an organism, if you remove a single cell from a multicellular organism and look at it,
does it behave differently?
Will act differently. It'll be in a different environment. Essentially, a multicellular organism, like us, we're just a big environment for our individual cells. Yes, it will act differently, which is why a lot of the time we sort of freeze the profiles before we take them out. So we'll just sort of take a snapshot. So I don't know. Sorry, I clicked my fingers. I don't know if that's a horrific sound on a podcast.
No, it's good.
Won't do that again.
It gives gravitas. Definitely. So then coming back to the single cell organism, it must be a lot easier to study them, or actually do you miss a load of variables that would help in the study?
Yeah, there's advantages and disadvantages. The advantage is you get rid of all that noise. What you see is real. It's real for that cell. That cell is living that moment. So that cell may be experiencing some sort of nutrient limitation. It may be experiencing some sort of environmental shock. But whatever you see is real. The downside is you're dealing with a single cell, so you haven't got much material to work with. And that's why you need sort of specialists in it, because you need really sort of high-tech equipment, which is sensitive enough to deal with one cell. and you need an environment to work in that is totally devoid of contamination, because as soon as you look at your sample, you're contaminating it. So, for example, the facility that I used to work in to do single-cell genomics was a clean room, which was not just a tidy, cleaned room. I mean, it had 7 HEPA filtration units on it, and had, I think it was about, last time we counted, about 15 particles under 0.2 microns in size per cubic meter. It's a sort of cleanliness that goes beyond microchip manufacture. And that's because as soon as you sort of have a single cell and you're trying to amplify a message from it, you don't want any contamination in there. So you go into like, it's almost like police forensics, but you cannot afford to make a mistake.
I'm really sorry for asking this, but the question, someone listening has got to have the same thinking process as me on this one. But what happens if you fart in there? Do you have to wear charcoal underwear or something to stop that from...
That's not the most obvious question of it, and I've never been asked that.
You're joking. That's the first thing that comes to mind in a room with 7 HEPA filters.
Funnily enough, I'll come back to the fart question. I'll answer the question and then I'll come back to farts, because that actually is a story from my PhD. But
right.
But in the room that you do this research in, you wear sort of non-shedding. They look a bit like crime scene investigator suits. So you look like a Stay Puft man, the big white decorators overalls. And for me, I don't have to wear a hairnet, but anyone with hair on their head have to wear a beard mask. But even then, you're still shedding. Even the bits of sort of skin that you can see are still in there. So as soon as you move into a single cell genomics facility, you contaminate it all. It's just about managing that contamination, working very quickly and smoothly, and sort of preventing yourself from damaging your sample. It's because a lot of people think that science is very dangerous, and like you see people wearing lab coats and glasses and things like that. And sometimes we do use dangerous chemicals and reagents and things, and that's why we have personal protective equipment on, But in this instance, we're protecting our samples from us, not us from our sample. So it's turning that on its head.
So do you wear charcoal underwear, charcoal packs?
There's no need. And, you can hold it in for a couple of hours.
It depends how much curry you ate the night before. Well, go on then, give us the PhD story on your farting. Well, I'm assuming it's your farting.
It's my boss, my PhD advisor, actually. We had a really in-depth discussion once on... So I did my PhD working on E. coli, which is an enteric bacteria that everyone has in their guts. And we were having a conversation one day about how disgusting gyms were. You think about, if you've ever been to a gym and you're on the treadmill and you're sweating, and you get that sweat running down your back and between your ass cheeks. And then, of course, it runs down your leg, really, doesn't it? And then you're thinking, well, you're just spreading enteric bacteria everywhere. And, if you think about these things, you'd never go out of the house. But, it's lovely now. I go to my local gym, and because of COVID, everyone's cleaning everything all the time. In many ways, we're sort of hyper obsessed with it. But we did get onto the topic of general hygiene, and one of the things is if you fart, there will be bacteria associated with that, and how thick do your underpants and your jeans have to be before no bacteria come through? And then we started a conversation about when you're wiping your ass with toilet paper. You think how thin toilet paper is, and you think, all right, when you're a student, you're using that sort of sheet stuff, and that's probably the best stuff. And then you sort of become a proper functioning member of society, and you're using double ply. That's quality stuff, but it's very fragile stuff. But maybe the best thing is the 3-ply luxurious stuff, because if you think about when you clean up anything in the kitchen, you have a tea towel or anything like that, you can feel what you're touching through the cloth. How many times do you have to fold that over before you're not in contact with it? I guess the end story is wash your hands after you wipe.
Well, to add to that, seeing as we've got to that stage in the conversation so quickly, I haven't actually used toilet paper in over 2 years here in Bali, because nobody uses it except to pat down and dry. You use hoses, right? And all of the toilets have these amazing upward jet systems with 6 different speeds, or you have a hand hose. And typically you're squatting and everything's wide open to exposure, and you spray the living daylights out. It's much more hygienic than paper. I go to the West, and I was in Singapore a couple of weeks ago, and I used a toilet there and had to use toilet paper, and it's kind of gross, Yeah.
Well, when you think about it, I mean, there are a lot of things that humans do that are pretty gross. I mean, the toilet paper is a bit of a weird one. I totally get where you're coming from. I mean, washing with water has got to be better than. Yeah, but I mean, the worst thing we do is, like when you think about what we do with our kids, and disposable nappies. You think, you take your, for those people listening to this who are lucky enough to have children and survive that, you take this wonderful being that you've created and lovingly nurtured, and you wrap it in essentially a piece of plastic, and when you change a nappy, you put a nappy on, and then 9 times out of 10 they go for a poo. That nappy's on for 10-20 minutes, and we take it off, wipe up, and then wrap this lovely turd in a plastic case, and then send it to landfill where it'll be preserved for 500 or 600 years. Our children's will have their bodies degrading before that plastic degrades. It's crazy. Yeah, that's what we do. I mean, and society has changed where it's all about convenience, isn't it? When do we get to that stage where that is acceptable? When you look at what we're doing, I think that for me with sort of plastic is crazy. I mean, plastic is vilified and said to be an awful material, and it's not. It's an absolutely brilliant material. It's just our application of it and our uses of it are disgusting. I mean, that's what we need to think about. We use plastic for single-use objects. That should never have been the case.
So here in Bali, we have a farm behind this office. We're working on regenerative organic practices. We're testing lots of bacterial fertilizers, all organic, in different ways with different soil. So we're testing those bacteria on the old rice paddy fields that have been sprayed with chemicals for the last 30 years. We're testing it with manure. We're testing it on straw. We're testing to see which works best. But we have a stream next to it. I say stream, it's about 1.5 meters wide, can flow sometimes 80 centimeters deep and quite fast. It's part of the Subak system, so we call it the Subak in Bali. And we have garbage traps in it because when I moved here, I started seeing the amount of garbage that was going down into the ocean, and it breaks my heart. I mean, I'm an unintended environmentalist, but I am one at my core. I can't see a piece of litter without picking it up. Difficult here because you spend your whole day just bent over. We put litter traps in, and I was really surprised to find on day one about 30 pounds of garbage overnight caught on the trap, and at least a half dozen disposable nappies. And I mentioned this to a friend here who has been working in this whole garbage solution for a number of years. And they said there's thousands and thousands of nappies every day that are thrown into the water systems here. And it's actually cultural in that there's a very strong belief in Indonesia, not just in Bali, that if you don't throw the diaper into the river, the child will get nappy rash. So it's this superstition. And I thought about this, and I thought that's ridiculous because these diapers are actually disposable diapers are a very new thing here. So how does a superstition develop a new one? Because typically they're going out of fashion, superstitions. They're not coming into fashion. They're from an older world. And then I realized, of course, that actually probably 30, 40 years ago before disposables, if you didn't wash the cloth nappies that the kids were wearing in the rivers, they were getting nappy rashes and they were getting probably infections and the like. So it's just this, it's converted from wash the cloth nappy or the child's arse in the river to now you have to throw this disposable diaper into it. And of course these things, by the time they hit the traps, they're these big, soggy, half shit filled. They're just the most gross. I mean, that's not we get human turds all the time in our garbage traps. And there were chickens yesterday because they would get sacrificed here. It's a real, you got to have a strong stomach to be able to stand in with your wellies and dig this out twice a day, actually. But we're talking about plastics, and I could talk about the amount of that's going on. But in your family, you're not actually the smart one, are you? Your daughter's the smart one.
Well, yeah,
isn't she enabling you to come up with new scientific processes?
Yeah, well, yeah, it's sort of, you don't want to sort of force your kids into science, do you, as a scientist? But you want to sort of, steer them in the direction if they could make it that way, and that's a nicely primed conversation starter. So yeah, we moved house about 6 years ago, and but I'd worked in a marine lab for about 14 years or 13 years at that point, but I'd always lived inland on Dartmoor, and then we moved to the coast for a sort of change of lifestyle, just so the kids could go to school with my wife, who's a teacher, and we'd see them a bit more. And so we long story short, we moved to the coast, and I suddenly developed a connection with the ocean far more than I had just working in a marine lab, because as a single cell genomicist and molecular biologist, I'm in the lab all the time, and a tiny little bit of volume is enough for me, So I bought a book to try and learn about the shoreline and learn a bit more marine biology, because I wasn't trained as a marine biologist, before I became a marine biologist. So and I bought a book on the shoreline. Then I got into seaweeds, and I'd always worked on microalgae, but before then, and I thought, well, I'll learn some science with the kids. So I'd walk along the beach with the kids, and we had a little. I've got the book somewhere around here, but we had a book on the seaweeds, seaweeds of the British Isles, I think it was called, and we had lovely stickers with hearts and smiley faces on. And when we saw a seaweed, we'd stick a sticker on the page and we'd tick it off on our list. And our aim was to get 10 seaweeds, then it was 20, then it was 30, and I think we're up to about 60 or 70 now, just from our local beach.
There are that many seaweeds in the UK? Yeah,
people just don't look. It's only when you start looking closely that you actually start seeing them. And it's sort of turned into a bit of a sort of family project now. And I started what I call the Seaweed Apothecary, which is we started sampling the seaweeds, and I took them into work, put them in a -80 freezer, and then freeze dried them. So basically preserve them without water in little jam jars. In fact, I've got some of those around here somewhere as well. I'll have to find those for you later. And it looks beautiful. Looks like, like a little lovely sort of spice rack. But it's become an amazing resource for scientists now. So people are always asking me if they can sort of get a couple of grams of this, a couple of grams of that for a scientific experiment. So I sort of share them freely, because ultimately it's cost me nothing to collect them. It's just an enjoyable pastime with the kids. And one of the projects we were doing at the time was on fuels, converting seaweeds into fuels. And my daughter was walking along, and she was getting annoyed because when you pick up lots of seaweed, quite often there's sort of plastic associated with it. We get a lot around here of old fishing gear, because we're just up from Brixham, which is a sort of fishing harbour. And there was lots of sort of nylon twine wrapped around the seaweeds. And my daughter sort of made a comment of kind of like, oh God, can't. And I was quite proud of her, actually, because she turned a scientific technique into a verb. And she said, Dad, can't we just hydrothermally liquefy this seaweed with the plastic? And I was like, no, you can't do that. That would be silly. I think she was about eight at the time. And I sort of got home and thought, actually, she might be onto something there. And I think I spoke to a colleague about it who did the conversions, and his initial reaction was, nah, it wouldn't work. And then he was like, but it might. So we just tried it out, and it worked beautifully. And we found that if we put the plastic in on its own, nothing happened. But if we put the plastic in with the seaweed, then it converted beautifully. So the seaweed was catalyzed in the conversion.
Yeah, carry on. But catalyzed into what? What happens
we were
to
using
the two components?
So we were using a process called hydrothermal liquefaction. So
yeah, you need to explain that, because your 8-year-old knows it, but we don't.
There. Just let me. You're always interrupting me.
Sorry. There's just so many points in this that I need to clarify.
So it's a process called hydrothermal liquefaction. So hydro means water, or wet. Thermal, using heat, and liquefaction is just basically sort of liquefy everything. And basically we would put seaweed into this container, pressurize it and heat it up, and then it would get converted back into an oil, basically. And you get an aqueous phase where all the nitrogen and phosphorus was. You get a gas, and you get a char, an inert char. And this is a process we were developing for fuel and fertilizer production from seaweed. And then we sort of started playing around with plastic in it as well, and found that, we were really lucky, actually. The first plastic we tried converted beautifully. After that, we tried other plastics, and they didn't convert so well. So it was one of these eureka moments that was sort of followed a couple of years later, when we actually had the proper funding to do it, where it was like, oh, it doesn't quite work as well as we want. But the actual idea came from an 8-year-old who said something naively that made us think differently, if you know what I mean. And I think that's a real problem, I think, with science sometimes, is we know what we know, and we know what we think we know, and sometimes we dismiss some really obvious things. And I think that's been a sort of theme in my career, really, is because I hopped around lots of different subjects. I don't know what I don't know, and I quite often ask the naive questions, and I take a stupid approach to things, and quite often that allows me to make breakthroughs that other people wouldn't, because they're not stupid enough to try what I would do.
So I've studied complexity. Yeah, it does completely. I've studied complexity science up to a certain point. I mean, I'm not a complexity scientist, but I've taught it as well in classes as it related to my previous business, which was training resilience in organizations. And Dave Snowden, who is one of the leading professors in complexity science, I've followed his work for many years, actually worked with his organization. And one of the studies that he quotes a lot is the gorilla in the lungs. Have you ever heard about that? So
no,
if you get I think it was 32 radiologists, specialists in looking for a very specific type of lung tumor. You show them an X-ray of the lungs, and you ask them to look for that tumor, and they look for it. They spend 5 minutes, and they say, no, it's free of that tumor. Great. Was there anything else there that stood out? And I think it's less than 30% of them will say, well, yeah, there's a tiny gorilla in the left lung that's been put in there. It's less than 30%, right? And so if you show the same X-ray to a bunch of people you just pulled off the street who've never seen an X-ray in their life, don't know anything about lung cancer, they all see it. They all go, yeah. Oh, yeah, okay, there's a gorilla in the lungs. That's funny, ha, ha, ha. And what the point of the study is, or the study is pointing at, is that the more expert we become, the less likely we are to look for novelty and to look for, Dave calls it side casting, which is looking for adjacent possibles rather than very linear, like this has to lead to this, this to this. And I've seen that with a lot of experts, that they just can't see the cliche outside of the box. And the box is often very tight. It sounds like you are very good at side casting and seeing gorillas.
Yeah, I'm probably more better at being a layman in different scientific areas, because I don't, I naturally see the gorillas because I have no idea what I'm doing half the time.
I'm sure your students are going to be absolutely thrilled to hear that.
They'll survive. They kind of work it out already.
I think that's the first. You know, I think that's the first quality in maturity is being able to acknowledge that you don't know what you don't know, right?
Yeah, exactly. I mean, I'm developing. I have a sort of student-led project that I think I've had about 4 or 5 different students work on it over the last 15 years, called Rumsfeldian Science. And I give them all the quote from Donald Rumsfeld on the known unknowns and the known knowns and the unknown knowns. and I try and get them thinking about a little project that I've been working on. So I know everything about this one gene that comes from this virus that I've been working on. I know where that gene is in the world. I know when it's expressed. I know where it's expressed inside cells and seasonal variation. I've taken that gene, I've cloned it, and I've expressed it. I've made the protein. I've crystallized the protein. I've made 3D printed models of the protein. I've held this protein structure in my hand. I know everything about this thing. I know where every atom is in the protein, and I know every nucleotide in the gene. I have no idea what it does, though. So as a philosophical debate, is it a known unknown, or is it a known unknown, or is it an unknown known? It's definitely not a known known, because I don't know what it does. And I definitely think it's not an unknown unknown, because I know too much about it. But it's an interesting field. They always come to a different conclusion, and they still don't know what it is, and I still don't know what it is. So how do you publish that?
Well, it did stop Rumsfeld using that as for an excuse to invade Iraq, did it? So
yeah, no, they did. But maybe there'll be a use for this protein that'll come good.
I think it boils down to and again, this is my layman's understanding. I've probably spent as much time in science in the last 6 years as most people who do a master's degree and then don't do science beyond that. In it, I've been surrounded by and working with scientists, and it seems every time is that at the end of the day, fitness or utility beats reality. You can know all the facts, but if it doesn't work in the real world, if there isn't something that is beneficial from it, so what?
Yeah.
But I guess that's difficult to say to somebody who's a researcher who researches for the sake of research. You're actually doing it for practical purposes, is my understanding.
Yeah, we're very, very applied in my group, and we're I actually take a very creative academic approach, but with an industrial mindset. So we try and fail fast, and I think that's something that a lot of academic scientists fail to do. That there's a challenge. There's an intellectual vanity over knowing more and understanding. And I mean, this is one thing I admire about a lot of my colleagues, is they will hammer their head against that brick wall until they knock it over. And that takes a certain person to do that, type of person, and that sort of tenacious spirit and attitude. You can devote your life to something, whereas I'll bang my head against a brick wall and then just sort of shuffle around the side and walk to the left of it, or find a sledgehammer or something. It's a different approach, and that works for me. It doesn't work for everyone. I mean, my head of department said, "It's great having you in the department. I'm just glad that you're the only one. We can't have too many people like you." And I totally get that. We can't all be the same. My approach is very creative, but it's also very disruptive. It terrifies some people. We got involved in some electrochemical processing of liquids a few years back, and basically we apply a current into water and make microbubbles, and then we use the bubbles to separate out the algae that were in the water. We're trying to develop low cost, low energy separation technologies. And I walked in, and the postdoc who was working on it at the time, really talented, bright chap, I walked in behind him and said, "Reverse the polarity," just because I've always wanted to say that, because I watched a lot of Star Trek as a kid. And he's like, "Do you even know what that means in this context?" I was like, "No, not really." I leave all that to you. He understood the whole system. He knew the equations. I went, "Just reverse the polarity for me." He was like, "What do you mean?" I was like, just flip it on and off and put it in reverse." And he was like, "All right." And then he did it just to shut me up, basically. And we found out that something really interesting chemistry happened in the system. And we sort of lost. So what we were trying to do was separate out the cells, and we were doing that really well. And they were sort of floating at the top of the liquid. But when we reversed the polarity, when we did it, we killed a few of the cells and they broke apart. And the action of just, a tiny percentage of the cells breaking apart stabilized the flotation of the rest of the cells. They basically banged it up. And so it was an amazing phenomenon that we saw. We were like, hang on, that's brilliant. that we've just discovered something really cool there, just because I said reverse the polarity, and we went away and thought about it. I was like, what's happening here? And then sort of the guy solved it and said, look, it's cellular material, it's proteins that are binding up and stabilizing the flock. If we don't need to reverse the polarity, but we can add something in that will actually mimic that effect. So we need a cheap, bountiful source of protein and fats. So in the end, we were just adding a sprinkle of dried milk powder into our cultures, and we could stabilize and separate the cells out really, really easily and cheaply. It was taking seconds, whereas before it was taking minutes. So just from having fun and not really understanding what was going on. At the same time, you have to be really, really careful. In that you don't sort of do something dangerous, which is the downside of that approach. So we always have to have health and safety and risk assessment in mind when we do these things.
But what comes to mind there is companies like Google, who give their core money-making systems have to be extremely clear and extremely methodical, and they're churning their money from their ads and from their data collection, their data cells, and that just runs as the big machine that it is. But they also encourage their staff and give them time off for creative ideas. And you're the first person that I've actually ever heard say that fun is a benefit to science in this way. Typically, and I'm an outsider looking in, it is as I wouldn't say boring, because it can be fascinating for many people, but it's extremely straight, it's extremely square, it's extremely methodical. Whereas so many scientific discoveries have come from eureka moments, right? They've come from serendipity, they've come from mistakes, they come from not cleaning out the culture, and all of a sudden you've got mold growing and penicillin or the likes. So maybe you should start a new campaign of novelty and fun and disruption in science.
Yeah, like I say, you can't have everyone doing it because it's very disruptive, and it's not very. Like I said, you have to be very disciplined and fail fast. You can only have. You can only keep doing these things, and to a certain point where you think, right, I need to commit proper time and resource and energy into this. Is this a goer or not? But there's nothing wrong with just messing around in an area and just sort of scoping out the landscape, essentially. There's been, but people think that it takes a lot of money to play. It really doesn't. You can do science very, very cheaply, and you can do creative science very cheaply. We make bioreactors and stuff like that with, bottles and gaffer tape and stuff. The point is it needs to work once so we can work out whether it's worth investing in it properly. And I think that's an approach that is possibly being lost in science because we're becoming too specialized too soon, and we're sort of not. The people going into science now, it's all about impact and measuring and metrics and putting numbers on things. You can't put a number on creativity. Most of it is going to go nowhere, and there's a lot of pressure on scientists to deliver now, and I think a lot of people don't feel they can play because they haven't got the time, and I think that's really sad.
You need the backing of someone like Bill Gates, don't you, really, to be a good scientist?
We've had the backing of Bill Gates before.
No way. Tell us about that and how you met him. Wow. What a coincidence.
I think you've got a primer sheet, haven't you? Yeah, it didn't go brilliantly, to be honest. It was very interesting working with them. So we were working on a biofuels technology. Like I said earlier, the dewatering technologies, we were working on a high throughput liquid processing technology, which sounds really complicated, but it was actually really, really simple. It was a propeller in a tube. And when you put a propeller in a tube, you create a vortex. And as everyone knows, if you want to mix up or do any reaction, you mix it. You stir a bowl, like when you pull the plug on a sink, you get a vortex forming naturally. It's nature's way of mixing things very efficiently. And we were using this technology in what we call a vortex bioreactor to try and make biofuel. So we put microalgae in solution into a vortex bioreactor and then use the vortex to break apart cells and do the transesterification reaction and make the lipids, and then use the property of the vortex, which is throwing the light material to the center to take the lipids out in a sort of in a suction manifold. So it's a one-step process for liquid biofuels that was going to revolutionize planetary biofuel production. It never went anywhere because there were about 28 technical challenges which we met, but just not in a line. We can never get it working. But one of them was, could you just take a cell and break it apart? Simple thing. Could you take a cell and break it apart in a vortex? And we were working, like I said, we were working on microalgae at the time, which to get the lipids out of them. And we just couldn't grow enough algae. So you can grow bacteria overnight really simply. So we started using E. coli as a proxy, just as any old cell. And we put E. coli in and we were trying to break it apart. And my another awesome, talented postdoc, that I'm very much standing on the shoulders of my team when I present all this work. Another postdoc who was working with me said, well, we could put copper powder in there and use the copper to break the cells apart. But then we were worried about copper toxicity, because then we couldn't get the copper out. So we put the copper in a seaweed extract, alginate, which is also used in food fillers. So we had copper powder inside a little alginate bead that we were making. These beads are about 3 or 4 millimeters across that we used to make with a syringe, just pushing it out. And we put them into a vortex bioreactor and we killed the E. coli within seconds. And we're like, yay, brilliant, job done. We ticked that box off. Now let's go back to the algae. And then we were like, hang on, that's kind of a really cool observation that we could kill E. coli. And then we thought, well, would this be better as a sanitation technology rather than a biofuel production technology? So we wrote a 2-pager to Bill Gates in his Grand Challenges proposal scheme. And we got $100,000 to test the idea out. And it worked. So we went for $1 million, but they said, oh, you worked out a bit too fast, so we'll give you another 100,000 to do something else with it. And then we went through a few rounds of funding with them, tested the vortex bioreactor out on 4 or 5 continents. I mean, we North America, South America, in Europe, where we were, of course, South Africa, India. Our system went around the world and became this sort of really cool technology for basically sterilizing wastewater. It's pretty cool. It's not been commercialized yet. I mean, when I say commercialized, it's not been industrialized yet. We always said we'd give it away for free for humanity, for the good of humanity. But unfortunately, we went so far down one route, and the first key step we were stuck into with our research. We need to go back and redo it all. But the potential is there. It's an amazing, amazing kit, especially in the developing world, where all you need is a pipe and a propeller.
But is it just the vortex, or is it the copper being added as well that kills the E. coli?
It's that was the elegant thing. We never really knew how it was working so well. So the vortex means that you're getting massive turbulence and massive sort of contact. When we put the copper in on its own, it didn't work as well as it did when it was embedded in the alginate. So what we actually think, our theory, which we have very little evidence for, but I think it's a pretty damn good theory, is the copper, when it's embedded in the alginate beads, is acting like it's in a dry environment. So metallic surfaces, like all our door knockers used to be brass and things. You never get any bacterial contamination on those because the metals are, bactericidal. Now we have plastic everywhere and we have contamination everywhere, but metallic surfaces are bactericidal. When you get them wet, you lose that bactericidal activity. So, it drops down. So what we actually think is that it's mimicking a dry environment, but in an aqueous environment, and we mix that in, combine that with the mixing of the vortex, and you're just getting absolute crazy kill rates. I mean, we even tried it with Deinococcus radiodurans, which is it's known as Conan the bacteria. It's the bacteria that lives on the side of nuclear reactor cores. This thing is indestructible, and we could absolutely tear this thing apart within, I think it was about, for Deinococcus, it was about 20 seconds, whereas E. coli, it was half a second or less. It was absolutely amazing result.
Why is that not being used all over the world to prevent communicable diseases? Diarrhea?
The program we were in with the Gates Foundation was, we were funded in the Reinvent the Toilet campaign, which they have all the best names. So for them, they were about creating value from feces, basically. And, they had processes to create fuel from it, fertilizers, just do something useful with it and valorize sanitation, essentially. Our technology dealt with the wastewater that was coming off. If you want to do anything intelligent with poo, you need to get rid of the water first. The water is crap for processing things because it makes everything bulky and inefficient, very energy intensive. So you have to get rid of the contaminating water, which is where our technology fitted in. So that's why I think the Gates Foundation funded us, is that we were a sort of one size fits all technique for processing the water. But then they went down a different route and we sort of lost momentum there. And I went back to being a marine biologist. But we published it all. I think we put about 4 or 5 papers out on it. And people still get in touch with me about the Vortex bioreactor. And I sort of send them all the plans and stuff. And I hope that someone in the sanitation sector picks up on it. But it just needs, you have to understand that the water industry, especially in the developed world, is making so much money that there's very little push for innovation and change, because why would you change anything that's making you so much money? So that, there's a lot of politics involved there. I think there's a lot of sort of virtue signaling, we want to develop innovative technologies, but in reality, what's changed in the UK in water processing for the last 100 years? We're still using Victorian sewer systems and reservoirs that are hundreds of years old. Nothing is really changing in the water industry. That's why we thought we could make more of an impact in the developing world. But it's just really difficult when you don't really know the field that well. And like I said, I'm a sort of an imposter in the sanitation field. So what we really need is that sector to pick up what we've done and run with it, really. And I hope they will. It's all out there, freely available for them to see.
I'd love to see that. After this podcast, I'd like to see more on that, because I might have a few fits that we could put that in front of.
Cool.
Did you ever meet Bill Gates?
I did meet Bill Gates briefly. It didn't go particularly well. I'd flown to Seattle for the Reinvent the Toilet fair, and we were very much on the periphery of the fair, because, like I said earlier, we weren't a technology that was really addressing their problem. We were a technology that enabled the solutions that they were trying to develop to work in the real world. And I was a little bit disappointed that he was only spending about half hour at the fair as a walkthrough, and he was going to the 3 main people that were winning prizes. I think they were from MIT and Caltech and places like that. Some really awesome stuff on display. But I thought, well, look, I've just flown thousands of miles to be here. The least thing he can do is have a photo by the Vortex bioreactor. And he was sort of walking by on his way to see the people that he wanted to see. So I sort of stepped in between all his people and shook his hand and said, "Can you come over here and have a look at this?" And I suddenly realized that I'd sort of created a security risk. And all the people the previous day who'd been asking me about my technology, they weren't really interested in the technology. I think they were security people who were vetting me as a security risk, because all of a sudden they sort of moved in to protect it. Whereas I'm in the middle of this sort of security huddle, still shaking his hand at this point. And I said, will you come and have a look at this? And he said, I'll come back later. And I was like, okay, you make sure you do. But he never came back. That's not his fault. It's not his fault. But yeah, the chap who was walking behind him found it really, really funny. And he stopped to have a chat. And it turns out that he was the Prince of Orange, who's now the King of Holland. So he said it was one of the funniest things he'd seen. The kerfuffle that I caused.
It's a shame. It's a shame you didn't have this shirt on at the time, because he would have appreciated it even more.
Well, look! I tell you what. Look, I've got an algae here that you might like. Look, hang on. So this is.
Oh wow! Look at that. Is that a red? That is an actual. Why do you have algae next to your computer in your home? Is that to show me? Please tell me it's not something you just collect at home. Are you in an office space or right now?
Well, sort of. COVID sort of changed everything for me. I have an office at home now, but we don't have a spare room, so I'm actually in the corridor. So that's the toilet. And that's
Got it. So that's where you store the algae.
Yeah, well, that's not just on my windowsill.
Tell us about the algae, because it's something that you're big into, and I've seen a number of videos showing the benefits of spirulina and algae for cleaning out pollutants and waste spills and potential energy and potential biofuels, but none of it seems to be scaling. None of it seems to be really working as a solution.
Yeah, well, this algae is actually just looking at the name on it. Rock pool isolate. This was isolated by my son from our local beach, and actually,
what's he 3, 4 years old?
He's 11 now, but he was he was 7 or 8 when he isolated that.
All right.
So we were down at the beach, and there was a rock pool that had become isolated, and it got very warm, and it had grown algae. And he came running over and went, Dad, there's a really cool alga bloom in there. So we had a look at it, and I thought, yeah, this looks quite nice. And he was like, come on, let's go and have a look at it in the lab at home, because we've got a microscope here. And so we looked around, found an old Coke bottle that had been washed up, opened the top, rinsed it out, took the water, and then I took it into the lab, and then another talented postdoc of mine separated out all the different algae in there, and we grew about 30 or 40 of them onwards. And this is one that's really sort of very, very robust, very hardy. So it starts off, it grows green, grows like a really deep green, and then as it gets old, it produces a lot of pigments, carotenoids. It's a strain called Dynamiella salina. So it's a beautiful algae. So it's grown all around the world in salt ponds and things like that. So it's very salt tolerant, which is why it was grown here.
Where does it get its energy from to convert from green into carotenoids?
So it's photosynthetic. So it's taken just like plants. So it's grown with sun's energy, and it's scavenging nutrients from the environment. And yeah, it's a beautiful algae.
Does it not need carbon dioxide as well? It's in an enclosed jar, isn't it?
Yeah, that's actually, if you look at the top, it's loose. So there's enough air transfer in there to get in. So they don't need much.
So why algae? What's the benefit? I mean, for the person who's at home, who's like, what's this scientist on wasting his time with algae?
Look, you should have started with this.
What's the potential for it?
So I've been working on algae for about 20 years now. So microalgae are the sort of unsung heroes of the planet. So, people always think that the oxygen they breathe is produced from the trees, in the Amazon. But half the oxygen we produce that's produced on the planet is produced by algae in the sea. 70% of the planet is ocean. It's there's life in the top couple of 100 meters, photosynthetic life, and they're producing all that oxygen. So, next time you go to the gym and you're out of breath, every other breath you take, that's oxygen that's coming from algae, and you didn't even appreciate that. So they're the unsung heroes. They're sort of the lungs of the planet, the real lungs of the planet. And I actually, academically, the work I do, I work on viruses that infect them. So I take that a step further. So once you open your eyes, people's eyes to algae, then I open their eyes to viruses, and they're even better. They're just awesome to work on. So viruses, if you take any water, seawater, a milliliter of seawater will have 1 million viruses in it. It's absolutely staggering. There are 10 followed by 31 zeros. That's 10 to 31 viruses in our oceans. And I I was saying earlier, my wife's a teacher, and I sort of zoomed into a science lesson for primary school kids during COVID. And one of the things I did was I got them to write down the number of viral infections that occur every second in the ocean. And, you know, these are little kids. So I said, look, write a number 1 and then write 21 zeros after it. You know, if you write that number down, that's the number of viral infections that are happening every second in our oceans. You realize that, you know, it's a microbial world we live in, but actually it's a viral world. These things are killing on a massive scale. And without that sort of constant cell death, all of our global ecosystems would sort of shut down. And basically, viruses are the ultimate nutrient mover. They're constantly cycling things. So when populations get stale and stagnant, that's when you get death and destruction, you know. So what you want is all the globe and the planet to constantly be living and dying. And that's what life is. And without viruses, it wouldn't be doing that. So,
having just lived through my first viral pandemic, shouldn't you be a bit cautious about your your descriptions of how great the viruses are?
No, I shouldn't. Viruses can kill us, but without viruses, we'd all be dead.
So it's a kind of Faustian pact. We have to have it, even though we don't really want it.
We have to have viral infection on this planet. Without it, we would all be dead. Simple as. And that's awful that some viruses, you know, how many viruses can you name? You know, you could probably reel off 20, 30 different viruses. They all infect humans, right? So everything alive on this planet has viruses that infect it. Probably hundreds of different viruses. We live in a viral world. And I totally understand it's devastating when we get them and they impact on us. But actually, without their relentless activity, life as we know it wouldn't exist. I mean, mammals wouldn't exist without the acquisition of viral genes. So, you know, if you think about why are mammals so different, is, you know, we have our young inside the female body without any sort of protection. It's in contact with the mother's body. And you think, well, how does that actually work? Because it's a foreign body. Well, actually, humans acquired a gene from viruses called syncytin millions of years ago. Let's say a mammalian ancestor did that, I should say, not humans, millions of years ago. And that viral protein does what it does in a virus, which is downregulate an immune system so it can survive. Whereas human mammalian organisms have now taken that gene and are using it to allow their young to live inside them for 9 months. So it's amazing from that perspective. So although, you know, viruses get a bad rep, they're not all bad.
Your statement that we're dependent upon them, that's because they're breaking down a whole host of, like, everything from insects to the algae to plants to, and that decomposes, that becomes the nutrients for the next round of life.
Yep. Just everything's constantly growing. When nothing's growing, nothing's living.
And so viruses versus bacteria in that process, because I would have thought bacteria were that agent. I never really thought of viruses as being the ones that actually were the catalyst.
So why would so bacteria, you know, some of the enteric bacteria, they grow at they divide every 30 minutes. So why are we not inundated with bacteria? Why is it not planet bacteria? Something's got to kill them, isn't it?
Oh, the viruses are killing the bacteria.
Bacteriophage, yeah.
Are you serious?
So, of course.
What do you say? Of course, like everyone knows this, you know.
So there's a massive field developing in the 20s and 30s of phage therapy, but it was stopped when we had this magic bullet of antibiotics. You know, penicillin, you mentioned earlier, that we sort of serendipitously discovered. We developed all these antibiotics. And then before that, we were actually, especially in Eastern Europe, and a lot of Russian scientists were developing bacteriophages to treat dysentery and human infections. But when we had antibiotics, we sort of took our eye off that ball. We went all in on antibiotics. And here we are 70, 80 years later with antimicrobial resistance. It's a big problem. So what are we doing now? Well, we're actually looking to phages. So one of my old students, Professor Ben Temperton, he's awesome. He's got a citizen phage program running where they're isolating phages to treat human diseases. So that's the future. So the thing about
explain what a phage is before you...
A phage is a bacterial virus. It's just so phage is from the word to eat. That's when they first discovered them, they thought they were eating the cells. But essentially what they do is they get in, replicate, kill the cell and move on. But bacteria, I should say, viruses are very, very specific for the target host. Human viruses don't infect anything else. We get scared when we see sort of species jumps with the pox viruses, but they're very, very rare. So viruses can actually be used to target specific bacteria in infections in humans. So it's a really cool area of research.
Do viruses obviously replicate not replicate, but they mutate they mutate quite fast as well, don't they?
Yeah, and that's why they're so great. So one virus, when it infects a cell, it depends on the virus and the host cell, but you can make 100, 1,000 different viruses, and then of course they'll go on and infect. So you get a very rapid evolution. So we call it it's called Red Queen host dynamics. So in the Lewis Carroll books, the Red Queen says it takes all the running you can do to stay in the same place, and that's how you get populations interacting. So the host will increase in fitness relative to the virus, and then the virus will evolve, and you get a constant infection cycle where they're in synteny. So it's very, very cool. And ultimately, they're still infecting the same thing, so they're staying in the same place. But the viruses are evolving incredibly rapidly.
This is a legitimate question, trying to get into the granularity of it. But it's one I've never even thought about asking, even though I was told that I had chickenpox when I was, let's say it was 4 or 5. So I've known there's these things called viruses. I know there's these things called bacteria. What is a virus versus a bacteria? What are the differences?
So, definition of a virus is it's an obligate intracellular parasite, right? So it's not alive, technically. So you've got to consider a virus as a packet of genes, membrane bound, that has no metabolism of its own. On its own, it's just completely inert. And then it bumps into a host, whether that's a bacteria, a human cell, a plant cell. It gets inside, and then it basically is a parasite inside the cell. So it lives within the cell, becomes active, replicates itself, bursts out, killing the cell, and then infects the cells around it.
That sounds so morbid. I know you, as well as obviously it's all life depends upon it and the likes. But the bit that is difficult for me to get my head around is that it's an inanimate object until it hits life. Yeah. So, but when it's replicated, it has metabolic rate, it has respiration. Does it? It uses oxygen and energy and glucose.
Well, so think about a cell, any cell. But when it comes to a viral infection, it's a single cell that's being infected at any given point. And that virus is inside that cell. Effectively, that cell becomes a zombie. It's still alive, but it's doing the virus's bidding. So quite often a virus will get into a cell, and the first thing it'll do, it'll shut down normal host activity, and then it'll start expressing its own genes and turn the cell into what's called as a viral factory. And that's what it is. It's just a hijacked cell turned into a factory to make more viruses, entirely selfish. And then those viruses will go off and infect other cells and make more factories and more viruses. It's just a massive production line. Brutal, utterly, totally brutal and lethal.
I mean, it's
most of the time.
If it was in a Star Wars...
So I should say, so that's called a lytic virus. Some viruses can go latent, where they'll just think, oh, this cell is okay, I quite like it here. And then I'll just put my genome into the host genome and just wait until better circumstances. So I don't know if you've, like I tell you what, when you've been ill before and you think, oh my God, I feel terrible. And then all of a sudden you're like, oh, you look in the mirror and then you get a cold sore, yeah? And you're like, oh, that is sod's law, just what I need right now. Well, actually, that virus that caused the cold sore has been sat in your lip for donkey's years, probably. And it's getting the messages from the rest of your body saying, oh, he's terribly ill, he's going to die soon. And that virus is like, right, let's get the hell out of here. So then the cold sore comes through, which is so it can spread to other people. So viruses are cunning.
That explains why I've had cold sores at times when I've run ultra marathons.
Yeah,
right.
When you're stressed, when your body's giving out signals of. I love the way you just slipped in when I run ultra marathons. By the way, I didn't miss that. Yeah.
No, I'm not proud of them, to be honest. They were a period of utter stupidity. But any time when I've taken my body to its absolute maximum, climbing big walls,
when your body's at its limits, and it's it's giving off signals of I am incredibly stressed, the virus will pick up on that and just think, right, this body is incredibly stressed. Maybe it's best if I pop out of here and go somewhere else. So
that's a good signifier I'm doing it right, because I haven't had one in years. So, but I've also got a bit fatter around the midline, as well.
So the virus is enjoying it.
Much better living. I could talk about. I'm not just saying this. I could talk about viruses with you for hours because I've got so many other questions. To me, they seem like if they were in a Star Wars movie, they would be the ultimate expression of evil. They going into the host and they're replicating to just destroy and destroy and destroy. But there's this secondary benefit to them, which is that they enable life. There's almost this kind of poetic, inverse
I'd say,
beauty.
Yeah, viruses, I'd say they're not evil. I think they are the Force in Star Wars.
Oh, right. Okay.
So,
nice.
And the true power is when they're in the middle, really, when you've got the good and the bad. I mean, the fact is when they're infecting humans and then having a detrimental effect on our lives, that is incredibly bad. But in terms of the overall viral activity, without it with infecting the microbial world, like biogeochemistry would break down on the planet. So I guess there are always far more Jedi than there are Sith. There are only ever 2 Sith. They could be the sort of when the viruses go bad in humans, and basically the rest of the time they're on the light side with the Jedi infecting everything else. So
I've hit a Star Wars fan, haven't I? I found a Star Wars fan.
You have, yeah.
And we better not go down that rabbit hole. We'll have to save that one for another time. So look, nature. This podcast is all about also looking at solutions and giving people some level of hope about the fact that it ain't all just a one-sided slide into catastrophe with environmental degradation and planetary heating and the likes. You're in the game, you're working, you're looking at solutions. Where do you see, what are you most optimistic about right now?
Well,
oh, don't say nothing. Don't say viruses.
I personally am very optimistic because there are some awesome scientists out there who are putting their brains to the problems that we've got at the moment. Climate change is the big one, and I think most of humanity is sleepwalking into this issue now. People are, the COP meetings are great. The clue is in the name, though. We've just had COP 26. I mean, how many more are we going to have before proper action is taken? But at the same time, that there are some brilliant pieces of work being done to try and mitigate against that. And for me, as an algal scientist, full disclosure, I've recently set up a company to do this, which is set up it's called Seaweed Generation. We're going to do carbon dioxide removal using seaweed in the ocean. So that's what I'm really positive about. I think there's a lot of hope there. We're just getting started now. We're on the start of our journey, but I am very, very hopeful for it. Where over the last 20 years, my group has always sort of taken the approach that solutions, unfortunately, life is cheap. Human life in a lot of places in the world is deemed to be too cheap, especially in developing countries where, environmental destruction is allowed and poor human health as a consequence is acceptable. Big industry goes in, makes loads of money, and then it leaves a catastrophe behind it. You see that in mining sites with contaminated rivers, and big industry putting out toxins into the rivers and things like that. The Industrial Revolution has essentially done that on a global scale with carbon dioxide. It's a silent killer. People are now waking up to it. And at the same time, there are, like I said, the approach I was taking is that environmental remediation services, unfortunately, have to break even and pay for themselves these days. That's the capitalist society we live in, where if we want to save people's lives and improve their quality of living, we have to develop scientific processes that actually make it worthwhile to do that, which is really sad when you say it out loud like that. But if cleaning up a river, or if doing some remediation process can make a profit, then people will do it. If it causes the government extra money, quite often they won't. And that's not something I agree with, but it's something I have to work with. It's a constraint I have to work with it. So we've always tried to develop solutions that actually help out, as well as make a profit. So for the last 7 or 8 years, I don't know if you know much about the sargassum blooms that blight the Caribbean every year. So since about 2012, these massive algal blooms, seaweed, have swept into the Caribbean region. They decimate the beaches. Tens of millions of tons wash it up. Totally prevents tourism happening. It rots, it stinks, it gives off hydrogen sulfide. Turtles can't hatch from their eggs. It smothers coral reefs. It's an absolute environmental catastrophe. And I've been trying to make fuels, fertilizers, plastics out of this material for the last 7 or 8 years. Eventually, finally found the missing piece of my jigsaw with my co-founder of the company, which is carbon dioxide removal. So we're going to intercept this seaweed offshore, sink it to the deep ocean, and remove this carbon from the carbon cycle permanently. So that's what I'm really excited about. It's a really simple solution, and I think it's got great hope for sort of mitigating against some of this carbon dioxide that's in the atmosphere. So that's what I'm really positive about personally at the moment.
And just briefly, why are you focusing on removing CO2 from oceanic plants rather than land-based ones? Is there a difference in the quantity that you can suggest?
So that's a great question. If you remove it on land-based plants, you've got a tree. How long will the tree last? 100 years or so? And then we chop it down and burn it, and the carbon goes straight back up into the atmosphere. By using seaweed in the ocean and sticking it to the deep ocean, it goes down thousands of meters, and it's essentially removed from the carbon cycle. It's on a much slower rate. That's why we have oil deposits in our oceans. That carbon gets locked away for millions of years and gets changed into oil. So ultimately, we want our seaweed to be turned into oil over millions of years. But 70% of the planet is covered in ocean. That's the only space for growth we've got. Terrestrial land is incredibly valuable for our food production. You've seen how, bioethanol, I don't know if you saw the announcement where America's saying, well, we're going to stop creating bioethanol now from corn and wheat because of the Ukraine crisis.
Food.
Food. How much land can we afford to put over to biofuel production in the future? It's certainly not going to get any more, is it? There's always tensions on land for food and fuel things. To basically, for carbon dioxide removal, you need it locked in there. So that's not saying it's not part of the solution, but it's not scalable because there's just not enough land. And you have to remember the land is essentially a 2-dimensional environment. We work in the soil. The ocean is 3 dimensions. It's up and down as well.
One of the reasons we've gone after regenerative agriculture as a core activity to our business is because of the fact that carbon sequestering is obviously a benefit of putting microbes back into the soil, always having cover crops, taking a completely organic approach so you're not causing the microbes to actually excrete CO2 in a stress response to pesticides and urea and the likes. And some of the numbers I've been looking at is, farming globally puts out about 30% of the world's CO2 across all of the different industries within farming, which is obviously, if we could create a farming renaissance, and there's plenty of case studies to show that when you actually, even on a large scale monoculture, when you take a diverse approach to the soil microbes, you get these huge transformations in both crop yields, crop resilience, health, and CO2 sequestration.
I mean, if you look at modern farming practices, it's terrible, really, for the environment. Like sort of sterile fields of monoculture. That's not the way it should be. We're using massive amount of energy to fix nitrogen from the air using the Haber-Bosch process to create our fertilizers, which are then running out into the ocean and causing these seaweed blooms. That's why these seaweed blooms occur, is because we're adding excess nutrients to the oceans. Everything's out of sync at the moment, and we need to bring that back into normal cycling. And I think it is doable, but what we need to start doing is putting the environment first and not capital gains.
Absolutely. Economies require a planet. The planet doesn't require economies.
Everyone seems to obsess with growth. Consolidation, I think, would be a lot healthier for the planet. Everything can't keep on growing unless you get viral infections. Population breeds disease. The reason why COVID went global was because we live in a global society. Everyone's connected, and everyone's living so close together. That's why we're getting all these viruses spreading around the world now. It's different now. We're not isolated communities. We're all together and we're all growing. And that's why we talked about sanitation earlier. You didn't need intensive sanitation systems until we started living in cities. So what we've got is a very strange world. I mean, it's a brilliant world we live in, but we need some technologies to actually catch up with the lifestyle that we've created for ourselves.
I was going to end on seaweeds, but you opened one last door there, because I know I'm eating into your time, and I had an alert saying that our podcast time was over. But just one final one is that, as a scientist, as somebody who obviously knows a ton about viruses and the likes, do you see more pandemics on the horizon? Do you see that unless we restore, there's obviously a link between healthy environment and the ability of humans to resist viruses as well. Where do you see the future? Is there another COVID type?
I think that's a great question, which I could probably talk about for an hour. I think society at the moment is really, really interesting. I think we've all learned an awful lot from COVID. And I think if there was a new pandemic that hit, and there will be more viral pandemics, I can assure you of that. Virologists have been warning about this for donkey's years. It wasn't a surprise to any scientist, but COVID is the name was a surprise, but that's because it was. It could have been any virus, I suppose. What's interesting is human society is I see this in communities is we're disconnecting with each other. We're all online these days. We live in this online world. If you take that, you see what's coming next with the metaverse and things like that. People are probably going to plug in more than ever. If you see Wall-E, where those people are on the sort of space cruise ship and they're just looking at their screens. From a viral perspective, that's the last thing viruses want, because people aren't having the contact. People are losing the ability to interact with each other as human beings. So that will counter the spread of any viruses. That's really sad.
So COVID has triggered a behavioral change that will ultimately...
No, I think society was moving that way anyway. I think COVID highlighted it. So a lot of people thrived during COVID, being in isolation. A lot of people hated it. But people got used to it. So I think people are, regardless of COVID, taking that out of it, people are becoming more isolated now. We are seeing less and less of people. I mean, in the UK, most people don't talk to their neighbours anymore. if we say, all right, we're going to start working from home more, we're having less interaction with real humans. Everyone's pixelated. I met someone last week who I've been working with for 18 months for the first time in the flesh. That we are changing how we interact with each other. From a viral perspective, that's better in terms of pandemic management. I don't think it's great for human society, but it's an interesting side of that.
So, I've had in the last couple of months 3 people after they've reached out to fist bump me and I've shaken their hands, and they've either and this happened last week in the Philippines. One person who shook my hands begrudging and then turned straight around and used the hand sanitizer in front of me straight away. And I've had 2 other people recoil in horror at the fact that I'm now shaking a hand. And a couple of years ago, it would have been rude not to, right? It's all now fist bumps or bowing at each other. Well, we've never met, but I feel like we've just been down the pub and had a good old yarn. And in all honesty, that this conversation has gone into completely different areas than I expected it to, and it's been incredible.
Me too.
I would love. I mean, it's Friday here. It's 20 to 6, and I've got my sound recordist. My Anne's falling asleep over there in the corner. She's nodding off, and I've got kids waiting for Daddy for dinner. I'd love to get you back on though, because I haven't even started with my questions. I mean, I got all this. This happens every time, by the way. I got all these questions, and I never ask any of them because we go down these amazing, interesting, fascinating sidelines. So, thank you so much for being on. Where can people find you if they want to know more about your work? My
website's just being built at the moment. I've had to take it down to change some stuff on it, but in a couple of months it'll be back up and running, which is bluemicrobe.co.uk. Okay. Otherwise, just Google stalk me and they can find my work. If they put Mike Allen Exeter into Google, they'll be able to find me. Okay.
Are you on Instagram or TikTok doing dances?
Oh, I'm on Twitter as well, at Mike_J_Allen, which is right. Where it's at.
Because I was told on Twitter that you take no prisoners.
I think I'm quite nice.
That's what I was told. Grace, who's our comms director, said, he's hilarious on Twitter. He just calls out all these different people and he's just takes no prisoners on it. So I haven't actually stalked you on Twitter yet, so I don't know what.
No, I don't think I'm that bad. There's some brilliant Star Wars related microalgal art there that I constantly put out. The irony is I'm waiting to go viral on Twitter.
Hey.
All my virus tweets go nowhere. So
all right. Well, you're definitely going to go viral after this podcast, especially when we put loads of advertising budget behind it. We'll push it out there. Mike, it's been an absolute pleasure. I know we've got loads more to talk about. I'll put all your email in the show notes, not your email, your website and everything else. And I'd love to get you back on to continue the discussion.
Yeah, invite me back on whenever you like.
Great, but we'll different shirts next time. We have to keep changing the shirts up.
Sure. Yeah? I'll get a new one.
I won't look like a prisoner next time. I'll send you one. I'll send you one from here. Send me your size and I'll send you a barley made shirt.
Fantastic.
All right. Awesome.
All right,
thank you.
Great meeting.
Cheers, mate. Thanks. Bye.